Welcome to the new season of Data Skeptic. This time we're calling it Animal Intelligence. A little bit outside of my usual comfort zone, so I've invited Becky to come on and help me kick the season off. Becky, why don't you take a second and introduce yourself?
Thanks, Kyle. My name is Becky Hansis-O'Neil, and I'm a Ph.D. student at the University of Missouri St. Louis in Dr. Amy Dunlap's lab, and I study learning and cognition in bumblebees and tarantulas. And I've been studying animal behavior since I was a sophomore as an undergraduate. My career's taken some twists and turns, but now I study learning and cognition in bumblebees and tarantulas. And I'm super excited about animal intelligence and to help more and talk more about the series.
So you've been helping me find guests already, in addition to teaching me a few things about the field. I guess my first question for you is, what do bumblebees and tarantulas have in common?
They're both arthropods, so they are segmented invertebrates, so animals with no backbone and segmented bodies. So with tarantulas, they got their head and their abdomen, and with bumblebees, they have the head, thorax, and abdomen. So insects and arachnids, little branch there. And I actually didn't plan on studying the bees. I got to University of Missouri St. Louis, and there was a really geriatric old bumblebee colony. And I said, hey, I'm not doing anything yet. Can I play with those? And I made a little operant chamber for them where they crawl into an artificial flower, and I pipette in a little sugar reward for them. And I just fell in love with working with them. They're such a cool model organism, and they became part of my dissertation.
And can you expand on that term, operant chamber?
Yeah, so this is a super common method used in studying animal behavior, dare I say intelligence. I think it started in probably around the 50s. So most folks, when they learn about it, they see a picture of a rat or a pigeon, and they're in a little box, and there's a lever or two in the box, or if it's a pigeon, it's like a little lighted key, and the rats press the levers, and then a pellet drops into a little magazine, and the rat gets the reward. With pigeons, it's usually a grain hopper, and it's a little bit of grain. But you can build this for a bee. You can build this for most animals. The ones we're going to talk about today, we'll get to talk about, hey, there's some animals where it's really difficult to build an operant chamber, so you have to use other techniques. But yeah, for bumblebees and pretty much most mammals, I would say, easy to build. And that allows us to get rid of all the extraneous environmental noise and look at just how the animal is responding. And then we can slowly add new stuff in, like, do we want them to discriminate between two visual stimuli or stimulus and things like that? So you can make it real complicated if you want to.
I bet. Well, you'd use the word, maybe tentatively, the word intelligence. Maybe we should do some vocabulary. What words are you comfortable with, and what does your field usually use?
So I come from a few different fields. I have a bachelor's in psychology, and my background there is in behavior analysis. And the behavior analysts are really hardcore about making behavior a naturalistic science and only talking about things we can observe. So they might not use the term intelligence ever, even though they're working on cognition and things like that and how animals solve problems. And then the other part of me is a biologist. I have a master's degree in biology. I'm working towards my doctorate. And we're much more comfortable as biologists using terms like intelligence or cognition. Usually when people are talking about intelligence, I'd say colloquially, they mean problem solving. If you can make this problem solving flashier by using tools or language, even better. Intelligence is going to vary quite a bit study by study. So anytime you read a study that's about quote unquote intelligence, you're going to have to figure out how they're defining it. Is it problem solving? Is it the ability to have certain abilities like using tools, language, that kind of thing? Or are they not using it at all? A term you'll see really commonly in animal behavior is something called cognitive flexibility, because intelligence is usually something we ascribe to people. And is it really fair to ascribe that to a bumblebee or a cat or a dog or a pigeon?
And what about learning? I don't know if that's separate from intelligence, but is it fair to say we can say with certainty all animals learn?
I think that that's fair. Now, there's different types of learning. You have associative and non-associative, and within associative, you have classical and operant conditioning. So there's different levels, but I'd say if it has a nervous system, it's in the neuralia, as we say, it can probably do at least non-associative learning. So that's things like habituation. So if you have a sea slug, aplasia, and you poke I'm going to call them antennae, but I'm sure their eye stalks have some other name, and they slowly habituate to that, they stop retracting them, they learn that you're not really a threat. That's sort of the minimum, is that habituation. Or if it is threatening, getting the heck out of Dodge. And this is all reflexive. They're not like thinking about if something's threatening or not. Those two things are going to be the basics. And I'm pretty sure you're going to find that in all animals. Next would be classical conditioning. So that's going to be your Pavlov's dogs. So that's associating a stimulus, so something you're perceiving, with a reflexive behavior. So in Pavlov's dogs, that's the scent of food with salivation, and then eventually ringing the bell causes salivation. So you're associating stimuli together. And then operant conditioning involves voluntary behaviors. So that's usually, if people are looking at intelligence, I'd say that's where the meat, where the good stuff is doing operant conditioning studies. But you can mix and match and make it super complicated or not even worry about those terms at all and still study learning. It's a huge field that's taken up by lots of different disciplines.
Well, it's good to hear there's a lot of schools of thought and ideas in the field. I don't think it's down to like one perspective. There's so many animals and approaches and just the things we've touched on before. I'm curious if there are any primary topics you're looking forward to us covering this season.
Yeah, so I'm actually really curious to hear some of our guests that are out of my realm of knowledge, that are really more into hardcore data analysis and looking at how sort of natural computing and how learning about animal cognition influences how we tackle computing problems. So basically, how are we using what we learn about animals to improve computing, data problems, all that sort of thing, because that's not something I know as much about. So I'm really excited about it. And then this has been super fun for me because I've essentially got to do a literature review of people that come from all different backgrounds, studying all different organisms. So pretty much everybody I'm kind of excited about. I want to hear each episode.
It's good to hear there's a data-driven component to it all, and I've seen a lot of statistics as I've looked through papers that are familiar to me. I haven't necessarily seen as much machine learning. How does your community perceive the machine learning world?
So my career is a little bit interesting. I'm a non-traditional student, so I went away for 8 years to work in the museum field and then came back to science. They taught us inferential statistics. And some people, if they were really fancy, were talking about Bayesian statistics during my master's. So this is like 10 years ago.
Sure.
And now that I've come back and I'm taking bioinformatics and systematics and these other classes, machine learning is coming up all the time, especially with genetics. And looking into behavior, I'm seeing where these same tools are popping up, especially things like hidden Markov models, for example. So getting a little bit away from bioinformatics and more into behavioral ecology. If you want to study an endangered animal, you might put a radio tracking collar on them or a GPS collar. And maybe this animal's swimming all over the ocean. And all you're getting is time series data about maybe depth, about GPS points, lat long. So if you have some behavioral information and knowing the probability of what an animal's doing, maybe at one point you strapped a camera on it and now you're getting these behavioral data, but you don't actually know what the animal's doing. You just know where it is and how deep it is if it's swimming in the ocean. Can we use machine learning to infer what the heck that animal might actually be doing out there? So in behavior, we're getting some really cool stuff. There's also been really massive advances in neuroscience in terms of mapping neural connections. So I've been bumping into more research, which I admittedly understand less, but I'm really fascinated by it, where scientists are using animal models and like saying, okay, how does the rat hippocampus, the part of the brain that deals with spatial navigation and memory, actually do all these things? And can we copy what the rat brain is doing in an artificial neural network? So I'm seeing a lot of this reverse too, where the neuroscience is getting good enough where we can actually use these biophilic or bioinformed forms of artificial intelligence. And one thing I came upon recently with pollinators, and this goes back into Markov models, and that's just what I've run into a lot. So there was another student in my lab. Her name's Rachel. So if Rachel hears this is your stuff. And she looked at pollinators raised in urban, rural, and suburban areas, and she filmed their behavior on flowers and found that they had different orders of different behaviors that they did. So to me, that's really cool raw data, because she also looked at things like gene expression. We can look at things like body condition in the animals. So that is so much nicer in biology if you can just film what an animal's doing and infer how it's doing. Like, what's your body condition likely to be? So these hidden state things, this just blows my mind. I love it. And then lastly, big advance in animal behavior is when I started, you would video a behavior. You would talk with all your lab mates and make what's called an ethogram, so behavior list, and you would try to objectively define each behavior. And then a couple of you would watch the video, make sure you're all calling the same behaviors statistically, that you're really highly correlated. And then once you hit that correlation ceiling, we call that inter-rater reliability, then you can watch all the rest of your videos. So machine learning is really helping us automate this process. So using computer vision, things like that, to recognize that rat pressed the lever, that rat is grooming, that rat is sleeping, as opposed to having to humans do all these things, because it's really slow and tedious and it takes a long time to train people. So that's been a huge use of machine learning in animal behavior and like intelligence studies. So those are my favorite examples du jour. Hopefully other folks are interested too.
Well, a camera on a rat or even a tarantula makes a lot of sense to me. When I picture a camera on bees, in my head it's just chaos. Like, how do you make sense of that data?
So one example, if I'm understanding the question correctly, one example for bees is, let's say, and we'll see if I actually end up using any kind of computer vision for this. I don't know if I will. So I give my bees caffeine. So part of what I do involves giving them drugs. And I want to know if all these bees are buzzed on caffeine, are they moving around a lot more? Are there any locomotor effects? So if I'm filming a colony of bees, what I can look at is, say, pixel displacement and see, are the caffeinated bees, am I seeing more pixel displacement versus not? Because when you're filming 200 animals in one shot, it's very difficult to track single animals. You can tag them, you can do that kind of thing, but we can use surprisingly simple stuff. Like, I wouldn't rank pixel displacement super high on the list of fancy tools. But yeah, tech helps us there too.
You got to leave us not hanging on the caffeine. Do they work harder? What's the end result?
So it varies. So this is just preliminary data.
Sure.
I might be changing my tune once the study's done.
Fair enough. That's science.
But basically, I ask them how much they're willing to pay for a controlled amount of nectar or reward. And obviously bees don't spend money, but they do spend energy. So if they're out in the wild and they visit a flower to get some nectar or some pollen, it's possible that other bees have already drained that flower of nectar or eaten all the pollen. So sometimes they have to visit multiple flowers. So that's what I want to know from them. What is the most flower visits you are willing to pay for a single reward? So if I throw caffeine in the reward, I have some bees that will double their payment.
Wow.
And then I have a whole bunch where they don't seem to care. And then others that pay less, which that sounds a little bit like people, doesn't it, with psychoactive substances. So I need a lot more bees to see how consistent these effects are. And obviously, if you have a bee that's giving you double the payment for a reward, you need to test that bee multiple times to see how consistent that effect is. So there's lots of questions, but so far it seems like some of them, they're willing to work more.
Recently, the world learned of the power of artificial intelligence, a technology cybersecurity leaders have been leveraging for years. Now, as AI expands and evolves, those same security leaders are left wondering where humans fit into the next generation of AI-empowered security tools and solutions. Arctic Wolf, the industry leader in managing security operations, seeks to understand this question in the newly published report, The Human-AI Partnership. Access the insights of over 800 cybersecurity decision-makers in North America and the United Kingdom to better understand how organizations are weighing the benefits and risks of deploying AI tools. Uncover the biggest obstacles to turning AI and human engineers into a formidable team. Discover why the near-term benefits of large language models are being upended by a crucial flaw in the technology. And learn what the risks of AI tools mean for human practitioners moving forward. Get your copy of this report today at arcticwolf.com/dataskeptic. That's Arctic, ARCTIC, Wolf, WOLF.com/dataskeptic, all one word. arcticwolf.com/dataskeptic. You touched earlier on bees being rural, urban, or suburban. I could imagine a ton of other factors: species, age, gender, the list goes on and on, what they ate for breakfast. How do you do a controlled experiment under this seemingly infinite set of variables?
So for my bumblebees, it's a single species. They're in the lab. I only use the females. I measure how big they are so we can control for size. So in the lab, it's really good. In the field, it's much harder. You have to identify the bee. You have to know if they're male and female. Luckily, sexing them is not super difficult. And they have vastly different physiology. So the males, their big job in life is to be able to find females. So they often have really big eyeballs. The parts of their brain devoted to the visual system are really well developed because that's that's what they need to do, is they need to be able to sense females going by. Females, they're more interested in flowers. They got to provision offspring. If they're a solitary bee, she's going to have her eggs she's taking care of. If they are a eusocial bee or a social bee where they have a shared nest, they might be bringing back resources for others. Maybe they're not even reproductive. In the wild, it's very hard, and you have to be good at IDing bees, and there's lots of courses. Some of them are really hard to get into. Like, there's competition for biologists, like, I need to take the bee course. So it's hard.
Well, I ask you to pick out one particular paper we could have a discussion about for this episode. Can you share for the listeners what you selected?
The paper that I selected is Operant Conditioning in Antlion Larvae and its Impairment Following Exposure to Elevated Temperatures. And the reason I chose this is because antlions are kind of what got me started on the whole invertebrate thing. So as an undergrad, I was getting my degree in psychology, and I was getting a minor in zoology, which was great. It enabled me to get my master's and do what I'm doing now. And I took a field ecology class, and I knew we had to do our own project. I wanted to do animal behavior. Well, you can't do any vertebrate research in the United States without going through a welfare approval process. So if you're an undergraduate student in a class, that's too onerous. So you're often restricted to invertebrate animals unless the professor has like an ongoing project. So I picked antlions, and I wanted to know what factors. So let's talk a little bit about antlions.
Yes, please, yeah.
Antlions are a larval state, so they're not the adults. The adults are doodlebugs, if you've ever seen them. They look a little like dragonflies, not quite, but they're larvae. The antlion, they find sandy soils, they dig a pit, looks like a sarlacc pit from Star Wars, and they bury themselves and their little jaws stick out the bottom. And animals, frequently ants, that fall in, can't get out. The antlions also fling dirt at them and knock them to the bottom of the pit. Antlion grabs them with these jaws, and the jaws are pretty big. It's like half their body length for a lot of species. They inject some venom. They're not dangerous people, don't worry, but they inject venom in their prey, immobilizes them, and then they drag them under the soil and eat them. So I thought, that sounds cool. I'm going to study antlions. And another student had already done it, so I knew they were at our field site. So I say, okay, I want to know what it is about their pits. Is it the soil type? Is it how big they are, how deep they are? What is good at catching ants? So I just put some sugary food out, catch my ants, go find my antlions, and I throw the ant in the pit, and I start a timer, how long before this ant gets away, how long before the antlion eats it. And then I measure the depth of the pit and the width, and I get a soil sample. Pretty straightforward. However, the antlions would fling a little bit of dirt, and then they'd stop moving. I thought, they're never going to catch any ants. The ants are getting away. Why aren't they grabbing them? So I tried a few more ants, and finally, my ant grabbed my antlion by the head and killed it.
Oh, boy.
So I thought, this is not what I read about in the antlion storybooks. This is not how this was supposed to go. So I thought about that at camp that night, and I thought, maybe my ants are too big, because they're about as big as the antlions. So I went and found some smaller ants, and the next day tried the smaller ants, and that worked. And the antlions immediately started flinging dirt. And I thought, wait a minute, how did they know? How did they know to hold still for the big ants and pretend, like play dead or whatever they were doing, stop moving? And how did they know to really aggressively attack the small ants? And that got me interested in learning in invertebrates. So I continued on studying beetles during my master's, and now I study, the tarantulas and the bumblebees. The antlions started it all. So I was really happy to find a relatively recent paper where they were looking at what we're talking about, operant conditioning. I would argue these researchers already demonstrated that antlions could do operant conditioning, that they could learn that well in some of their previous work, but they weren't so sure. We could talk about that as we go through the paper. But I thought this was a really good one, because this shows an animal that you can't really build an operant chamber for. Antlion's not going to press a lever. Their whole natural history is just sitting in a pit. So how do you test learning in an animal like this? So I thought that would be pretty good. And then they did some kind of interesting things with their statistics. I'm not a biostats guru, but there are some things they did in here that I'm seeing are trends in behavioral papers. And they did a good experiment. So, and it might surprise people to learn that, insects learn operant conditioning. Yep, they can do it.
Is there a floor to that? Does any, do bacteria learn? I didn't know that insects necessarily did either. I knew pheromones were at play. It seemed to me they could have been mechanical. Is everything learning?
I'm not a microbiologist, so
Sure.
I will give this explanation for microbes the best I can. They do have what we call behavioral plasticity, so they can change their behavior in response to the environment. It's not the same as learning as an organism with a nervous system, so it's sort of like oh, if I get chemical input on the right side of my quote unquote body, I need to move to the left. But there's no nervous system processing that. It's more like, yeah, what you were saying. There's a chemical input that's causing mechanical changes within and metabolomic changes within the organism, and it's responding. They can do things like that. Tends to be what I would call a little more hardwired. It's less flexible, but I know they can do some really surprising things with just chemical, basically, responses and things without a nervous system. So I wouldn't put it up there with the antlions, but it's surprising. I would be super excited to see if we ever are able to find extraterrestrial life if their learning system physically is anything like ours.
Well, the paper presented evidence against a common perception. I didn't know about this perception before, but apparently there was a perception that ambush predators, like the antlion, are limited in their cognitive abilities, or at least that foraging insects were somehow smarter. And maybe there's evidence now that that's not true, but do you have a sense of where the debate has its roots?
Oh, yeah. And spiders are beautiful. Tarantulas are a wonderful example of this. If all you need to do in life is sit in one spot until prey walks by and grab it, there's not a lot of cognitive demands on you.
I'd get bored myself. I'd have to do something, but yeah.
Yeah, it might be good if you don't have a lot of cognitive demands for your welfare. So you need to do less. And nervous system tissue isn't cheap, and it's usually metabolically expensive. So the body has to spend a lot of energy to maintain it. There's this idea that it's costly, and evolution is not going to favor it unless it's really necessary. So if you have ambush predators, and I'm not going to pick an insect, I'll pick a spider, like a wolf spider or a jumping spider. We see these really well-developed visual systems. They're not making webs that they just sit in necessarily and wait for prey. They're actively pursuing and having to outwit their prey. So the thought is natural selection is going to favor better cognitive flexibility, more learning abilities. Oh, I don't like saying that. But in these animals that need to hunt, essentially, and that are moving around a lot.
And then also I think we should cover the T-maze. I guess that's pretty commonplace in your world, but for listeners that don't know, what the heck is a T-maze?
If you want an animal to make a binary decision, a yes or no, you construct what's called a T-maze. So it'll have a shared, what we call arm, that you put the animal in, and then usually there's a left or a right turn. Sometimes they're at angles or do different things. And basically the animal has to learn to go left or to go right. And these are pretty helpful if you have an animal that's not going to be able to participate in an operant chamber. So antlions are good. A lot of the learning work in tarantulas has been done with T-mazes. You can make them really complicated and add more arms, and then they're called radial mazes. They get pretty fancy, but in this case, it was super simple. It was just a plastic T that the researchers could see into with little cups of sand at the left arm and maybe a cup of sand on the right arm. It depended on what the antlion did. And the antlion, essentially, they don't want to be out in the open on a smooth surface because even though they're venomous, they're very tiny. Their jaws are not going to pierce the skin of a vertebrate. They are good food. And when a lot of insects are in their larval stage, they're very fatty. They're a lot of lipids. So they're good things to eat. So antlions, they don't like that. They're not going to want to stay out in the open. They're going to go looking for sand to bury themselves in. So in this case, the goal for the antlions in the T-maze was to go left or right and to find their home.
At one end, I guess, is some sand that they're looking for. What's at the other end giving them the negative response?
The pit of death. Well, it's not really death, but it was an open pit. So if they go the wrong way, they fall through the T-maze down onto a table, which is, I just can't imagine antlions encountering that very much in nature. Maybe if a bird picked them up and dropped them. So yeah, that's probably very punishing. So we had our T-maze. The antlions could go left or right to get to their home cup of sand that the researchers put there. Before they started doing any training or any testing, in the control condition, didn't matter. Whichever way the antlion chose to go, regardless of their bias, the researchers put their little home cup at the end of the T-maze. So they never had to learn anything in the control condition because they always ended up home safe. Only thing they learned was to walk down an arm of the maze. For the training conditions, if they pick the wrong arm, the arm that their innate bias, so let's say this antlion likes to turn left. If it turned left in the training group, it would just fall through a hole and land on the table, which kind of an unpleasant experience, so they don't want to repeat that. If they kept making mistakes, they kept going left, the researchers would put them back in, and they would keep repeating until the antlion got it right. They also had a little protocol where they'd block off the wrong answer, come on, buddy, you can do this, we believe in you. At the end of the experiment, the antlions in the control condition were going left or right about 50% of the time. The ones that had been trained, they fell and plummeted to the earth when they picked the wrong choice versus making it back to their home cup, were making the correct choice about 75% of the time. Reinforcement and punishment also have super explicit definitions in a study like this. So punishment is anything that happens that reduces the probability that the animal will engage in a behavior again. So I'm hoping that this is like music to the ears of people that are really into data and computation, like, yes, rules and probabilities. I like that.
Yeah, very measurable. I would say that. Yeah.
Yeah, and reinforcers, it's just the opposite. Anything that increases the probability that a behaviors are going to happen again. And it has nothing to do with if the animal likes what happens or not. So the biggest confusion I get is people, when they hear the word negative reinforcement, what they're usually thinking about is what we call positive punishment. So telling the dog, no, bad dog, don't do that. But negative reinforcement is actually taking something away from the environment to increase the probability the animal will engage in a behavior again. So it has these very specific rules. So in this case, nothing too fancy. We have positive reinforcement. They get to their little cup of sand and they're very happy and they bury themselves. Or we have positive punishment where they fall through the air. So they're not going to want to do that.
Well, I like the T-maze in that it seems like the baseline is 50-50.
Yep.
That hopefully insects don't have like a right-handed bias or something like that.
In the methods, they tested each antlion, I think it was 5 times, to see if they had a bias for turning left or right. And I looked into this, and these researchers studied this before. And I've seen this with other animals, so they would flip the antlions on their back and see if they rolled left or right to get back over. And they are consistent with themselves. So they do have a bias. So I've heard that called lateral asymmetry in animals, because we don't call it handedness. Antlions don't have hands.
Fair enough.
But they do have a bias, and the researchers controlled for that. They figured out, okay, does this antlion tend to turn left or right? And then for all their experimental tests, they train them to go against their bias. So if you're an antlion who likes to go to the left arm of the maze, cool. We're going to put your little bucket of sand on the right, and you're going to need to learn to go right.
And then I guess when we go to do the analysis, we have to compare it against that initial measurement, right? So it's not, did you go 70% of the time and 50% would be the control? It's controlled against the initial measurement. Is that right?
So sometimes researchers do that. In this case, it looked a little bit different to me. So they had half their antlions that received 20 training trials. They actually actively tried to train them to go to the correct place. And then others that didn't get that training. They just tested them at intervals with no training in between.
Ah, okay, yeah.
And those peak out at around 50% accuracy for correct responses. And the ones that were trained, that got the training, I think in their non-temperature changing conditions were closer to, I want to say 75. So they did better. Statistically significantly better. For the control condition, the antlions were placed in the T-maze, and it didn't matter if they went left or right. The researchers just put their little cups of sand at the end of the T-maze on either arm. So there's nothing for the antlions to actually learn. So at the end of the experiment in the control condition, they were going left or right about 50-50.
So that seems like conclusive evidence of learning, that they figured out when I go this way, I end up where I want. Is there any doubt here that I'm missing?
No, I think this is a super straightforward experiment. The only doubt I had is in that experiment they did with asymmetry. So in the previous experiment, one of these authors used vibrational cues because antlions are covered in little hairs. They're very sensitive to vibration. So they would give the antlion an ant. Antlion would grab it, happy antlion. They would put a little bit of sand in the pit as the antlion was eating, and then they would grab the ant with forceps and take it away from the antlion in this previous study. And then others, they would use the vibrational cue, dumping little bits of sand in the pit, only at random times when the antlion was eating, like a little bit before, a little bit after. So there's nothing to learn. Vibration provided no predictive information. So what they found is after multiple trials of this, if they poured in a little sand as the antlion was eating, the antlion would pull the ant down under the sand so that they couldn't grab the ant away. The question here is, okay, is this completely instinctual? Because if that's instinctual, then it's classical conditioning. It's not operant conditioning. I feel like they did a pretty good job. That doesn't seem like instinct to me. Because when they get vibrational information, they have to know if that's prey, not prey. So that's telling me that they're processing something about the information that's going to happen. They're going to remember it, and they're making a voluntary choice to pull that prey down. So I think they already demonstrated operant conditioning, but maybe you could argue with me on that, I think. So I think this was fine. And I think they did demonstrate it very well in this new paper. So I don't have major concerns. I would have been a happy reviewer had it come to me.
Sure. And it's all behavioral observational. We can't interview them. There's no MRI for these things. Is that right?
Yep. They're small enough that you could probably do some brain slices and do some cool confocal microscopy to look at their brains. There's even electrodes small enough that you could probably do some brain activity stuff. But yeah, that's expensive, much harder.
It also sounds a bit sci-fi. Like, do we really know what the neurons do, or am I underestimating the field?
I think I see what you're getting at. I've heard neuroscientists give this criticism to MRI studies in humans, because depending on the kind of MRI or CAT scan you're doing, you're measuring different things, whether this is electrical activity or blood flow, and what is the correlate of blood flow to what you're actually thinking.
Sure.
So if we're looking at electrical activity in neurons of antlions, how much can we infer about what's actually going on inside the antlion? And this is a philosophical debate that I think has been going on in neuroscience for a while. So I think that's a fair question.
Will be interesting to see. Now, there's another variable we haven't touched on yet in this experiment, and that's temperature.
Yes.
Which I wasn't fully expecting to come up. What was the temperature component?
Yeah, so if you can throw a climate change angle into a study these days, it's going to make it a little more of a hot topic.
Mm-hmm.
In this case, they redid the experiment, and some of the antlions had a marginal temperature increase. I think they called it benign temperature was their variable, and then one where it was a much higher temperature increase. And these antlions in the wild, they are from the Mediterranean. They like to hang out in the shade. They're not like out in the desert cooking like a real sarlacc in Star Wars. They wanted to see what was this learning procedure like for these antlions that are living in these really hot conditions and hot when they're learning.
So that makes sense that temperature can affect them. How does it end up affecting the learning, though?
The graphs we've been looking at, I've heard them called learning curves. The way they graphed them here is really interesting. So you could actually see their general linear model surrounded by confidence intervals. So they're learning, I don't know if they would call it a learning curve, but essentially you're looking at accuracy over time. The relationship in the antlions that were really warm was less steep. And I think it was only marginally significant. So basically what that means is when they're warm, they're learning slower. The probability that they're going to get the right answer over time is a little bit lower than their regular old lab experiment that they did first. So shallower learning curve, not as steep. Additionally, they looked at mortality, and I think the smaller antlions had a little higher mortality in these heat conditions, but I'm not sure that differed by temperature. I think the little ones just struggled no matter what. So they looked at a whole bunch of different things. And then they believe they tested memory as well. And our antlions that were in the high temperatures did not remember which way to turn as well as the antlions that were at marginally or benign temperature changes.
In a sense, I kind of relate to it. I've been in some hot classrooms, and I don't think I got those lessons as well as the air-conditioned ones, but maybe I'm anthropomorphizing too much.
I noticed they kind of left stress out, like emotional stress as one of the reasons. So they talked about actual metabolomic changes. So maybe their metabolisms are ramped up because they're ectotherms and they're warm. So the actual chemical changes in the brain associated with memory are also sped up. So they're just forgetting faster. So I believe that was one of the reasons. And they listed one more, but it wasn't emotional stress. It wasn't like the antlions were in distress. And it could be they had that in there and a reviewer asked them to take it out. I just don't know. We certainly know in other animals that if they're really stressed out, learning a discrimination task, which is what this is called, to go left or right, that's much harder. So I think stress could play a factor. And it's one thing to say insects can learn. It's another thing to say they have emotional lives that are affecting their behavior. They may have just been very prudent about how they were writing about it. But I think that's possible based on the studies we have, especially in bumblebees, that in insects may have, I wouldn't say it's like human emotion, but something emotion-like happening.
It's hard to know not being able to interview them, but I do know at least in humans, emotions to some degree boil down to chemical reactions in the brain. Do we know that they have the same mechanisms where it seems plausible, even likely that they have emotions?
Yeah, so some of this work comes from bumblebees. So we know they have dopamine, just like we do, serotonin, just like we do. They have some other neurotransmitters that we don't, like octopamine, which supposedly functions a little bit like epinephrine or norepinephrine. They aren't responsive to opioids, but they are responsive to alcohol. I suppose the opioid thing's a little nebulous. Bumblebees don't respond to cannabinoids like we do. So there is some commonality because we do all share a common ancestor, but then there's lots of things where they're very different. So one of the studies that I've read most recently that tackles that head on is by a fellow named Lars Chitka, who has a bumblebee lab, just wrote a book as well. What he did was basically induced pessimism and optimism in bees and then tried to manipulate that by giving them drugs that blocked dopamine receptors. So he would give the bees unexpected rewards. So the bees like, ooh, I got some sugar while I'm out here doing this experiment. And in some of them, he'd give them drugs that would block the dopamine receptors. And he found that they behaved more like the pessimistic bees.
Huh.
So we can manipulate them pharmacologically in similar ways to mammals and people. And that's one of the big questions I'm interested in is, do the bees know when they're on drugs? So I just got a grant from the Insect Welfare Research Society. It's a new group where I want to know if bees can use the feeling, if it exists, of being high on caffeine as a discriminative stimulus. So knowing if I've been taking caffeine, can I learn that when I'm on caffeine, only the blue flowers give me rewards, and when I'm not on caffeine, only the yellow ones give me rewards?
Fascinating.
Yeah, and philosophically, you're never going to be able to prove sentience in another animal.
Sure.
You can't technically prove it in a person either.
Mm-hmm.
So all we can do is experiments like the one I'm describing, where we just chip away at it. We get a little bit of evidence. Yeah, long story short, yes, insects share some of the same neurotransmitters and things that we do. And in so much that has been studied, so far they seem to respond in similar ways, but it's very understudied.
And so this, I think, pretty conclusively to my read, and also from what I'm seeing in some of your replies and whatnot, that these methods do support operant conditioning works on this particular species. Is that shocking, or is that sort of to be expected and just not studied yet?
To me, that's to be expected and just not studied yet. There's nothing going on with insects in their nervous system that precludes operant conditioning.
All right.
And it seems like every insect we study, we find it.
And how long does it stick for? I can't imagine you come back later and they're still going left in the mazes, but a day or two, what's a typical retention rate?
I think that's really going to depend on the animal and the situation. I know in the bees, depending on the studies that you're doing, they can forget pretty quickly within a few days or even shorter, depending on what they're doing. But if you take an animal like a tarantula, where a female can live for 20 years, their memories might be a lot better. So some of that's going to depend on how long is that information useful and how long should the organism spend energy maintaining that memory, that connection in their nervous system. So for a lot of animals, it's not useful for it to be very long, and for others it is.
And then the temperature angle is interesting. As you'd mentioned, it's kind of timely to tie in climate. And I don't mean that as a criticism. It's a path to funding and it's relevant. But can we take inference from anything there? Is it that as temperature continues to increase and they're not going to learn as well, we're going to have dumber antlions, or they're going to starve more often? What's fair to inference from some of this?
And I think they made a few good points in the paper about that. So they were looking for sand in this study. And within their small little home ranges, when they're out in the wild, they might occasionally have to move. Like they were getting tons of ants and the ants disappeared. So, okay, I need to move my place and maybe I need to move it back again. I need to learn which stimuli is a predator in my pit versus food, all that kind of stuff. So if they're not retaining memories as long and they're having trouble even acquiring those memories as quickly, yeah, I think the things you mentioned, like potentially not getting as much prey, not moving their habitat or their pit as effectively when they do need to move it, things like that. So it's really subtle. It's the sort of thing that builds up over a period of time. Like, does it eventually get too hot? So there's all these things that can happen before it's so hot that the animals just dies. The heat is lethal versus altering their behavior. And this is one of these examples where their behavior changes, but way before we have to worry about them dying.
One thing I noticed in the paper, or one thing I missed, is I don't know if it discussed exactly the sample size of how many they were looking at. Whether it said it or not, I guess broad question for you in terms of experimental design. I don't think, every ant is the same or every tarantula is the same, but they also aren't the unique, interesting snowflakes that people are in my mind. There must be a little bit that's more. I mean, ants are clones, right? A lot of these guys, there must be some similarities between them. And bigger is better, I guess, in terms of sample size. But how do you look at a challenge like that?
So I think in this one, it was like 20 in the control and 20 in the experiment. And then I don't remember off the top of my head what it was for the heated trials that they did. What you saw them do with their learning curves is they had the probability the antlion would make the correct choice and then these 95% confidence intervals. And they used a general linear model as opposed to just a straight up linear regression, though I guess you could argue that everything's a regression in the end. So, what I'm seeing in behavioral sciences is this move to graph things in a way that show data distributions, confidence intervals, standard errors, things like that. Violin plots, if you or your listeners are really familiar with those, better ways to show data so that it's honest. And operant learning studies we have notoriously low sample sizes because it takes a while to train an antlion to turn right. Or in my case, like I'm going to try to train bees to react to drug stimuli and use that to make decisions. That's not going to be a ton of bees. So what you're going to see, I think, throughout this season when we talk about some of these really complex behavioral studies is lower sample sizes. And it's worth discussing with scientists how they deal with that. Like I said before, I think one of the big things we're seeing is a push to transparency in data so that everyone can see just how strong these conclusions are, especially if you have a lower sample size.
I think that's the right path. Even I have some accolades for the paper and how specific they were about how the larvae were kept. They were in plastic cups. They had this much food and all that kind of stuff. So seems quite reproducible at a glance.
This would be something that'd be totally fun to give to an undergraduate and go, hey, here's the paper. We're going to go get some antlions because they live here in the United States. Depending on where you live, you can find them pretty easy. I did for my study. So I think reproducible.
So for you, you just had to go out and capture some. Is that the process? There's no like facility you can order them from? Or is it yours just more fun?
I see, occasionally I'll see them for sale through bug wranglers. I don't know the legality of collecting antlions and shipping them across state lines.
Oh, sure.
Yeah, they're easy enough to find. I'm going to say this with a caveat, especially if you're a student or a researcher, you always need to check with your local state authorities about working with any wild animal, insect or not. But for the most part, there's not too many regulations if you wanted to go collect some and bring them into a lab. So it's pretty easy to get them, and it's fun to do.
Well, any closing thoughts on this paper?
I just thought this was a pretty good mishmash of showing, hey, what is it like to study intelligence in a naturalistic way, introduce some terms associated with learning and T-mazes and things, and give our antlions a little bit of a platform. A lot of people haven't even heard of them. So, yeah, I think it was a fun paper. I hope you enjoyed it too.
Yeah, it was a big learning experience for me. I'm looking forward to more.